Dr. Jaffe, can you explain what is different about the LRA microtiter plate and how that helps with the LRA test?
Yes, it's a very interesting set of engineering and optical challenges. In the early 1980s, it was my goal to develop a test that would allow us to measure white cell responses just as they occur in the body. And that meant rethinking how you would do a cell culture, because a cell culture requires that oxygen come in and carbon dioxide come out, that the cells in essence breathe. And so it seemed to me that we would want to have an environment called a microtiter plate. Microtiter plate just is a technical term for many wells on one piece of plastic. So we would want to be able to put it on a microscope and look at the actual cells as they respond to the specific substance in each well. And it was an injection molding challenge because the bottom of the well is very, very thin. Barely the thickness of a human hair. It's very, very thin. But it's optically planar, which means you can go from well to well and refocus on the living cells and observe how those specific cells are responding to the specific substance that was coated onto that dish, onto that well. We use a very specific kind of optically pure plastic bead, which is then injected through multiple ports almost simultaneously so that the mixing occurs very quickly and the polymerization that follows occurs very quickly. And you end up with this unique, very thin microtiter plate. Now let me contrast it to the size of a standard microtiter plate, which is quite thick by comparison. And we also thought ahead about the automation, not just the visual optical reading, but eventually having a machine read. And so we designed it so that the plates can stack upon themselves. That is, they could be put in a stack loader and sampled as optimum conditions present. There's a beveled edge so that the plate is always known, even if you were blind, but you had sense of touch. You could orient the plate on the microscope for someone else to read if you were not sighted yourself. But we thought ahead about how to make sure that once the plates are made smart because they're coated, that the orientation is absolutely defined for each plate of its kind. And if you put two of these spine to spine, you get exactly the size of a standard 96 well microtiter plate. And there are systems that would allow our very thin plate to be read in their very thick system. And theirs needs to be thick because there's so many steps. Ours is a one-step procedure. Once we put the cell-rich plasma in and incubate near body temperature for a short period of time, the plastic itself becomes the environment in which the cell culture is done and the amplification occurs. So we continue to have a unique combination of amplification and cell culture in the form of a smart plastic, very thin, but very functional environment.